Hello, dear colleagues, dear friends. My name is Florian Lordick. I'm working in the University Hospital of Leipzig in Germany, and I want to give you a talk about the personalized treatment of gastric cancer, current status. Here you see my disclosures. Gastric cancer is a global health problem. From a global perspective, it is the fifth most common cancer in the world, ranking number 4 in men and number 7 in women. There were over 1 million new cases in 2018, and it was the third leading cause of cancer death worldwide. When we are talking about gastric cancer, we are talking about different biologies in different locations. The Cancer Genome Atlas classification defined 4 subtypes. The most common subtype currently is the chromosomal instability subtype, which can occur in all parts of the stomach, but has a certain predominance in the proximal parts of the stomach, including the esophagogastric junction. The genomically stable subtype can be found in all parts of the stomach, while the microsatellite instability subtype is most commonly found in the distal parts of the stomach, and the Epstein-Barr virus associated gastric cancer is most commonly found in the fundus and in the body of the stomach. The chromosomal unstable subtype is associated with intestinal histology according to the old Lauren classification. Many of these cancers carry TP53 mutations, and often there is activation of receptor tyrosine kinases and ROS. Some of these receptor tyrosine kinase activations occur mutually exclusive as amplifications, but also co-amplifications can be found. An interesting publication from the Singapore group of investigators that came out some years ago revealed that a subgroup of approximately 1/3 of gastric cancers is characterized by amplification of specific receptor tyrosine kinases such as FGFR2, KRAS, OKI2, EGFR, and MET. At that time, we thought that we still will have developed specific treatments for specifically characterized molecular subtypes of gastric cancer. Unfortunately, this did not work in the way that we wished it, and what we currently have in our hands is only HER2 targeted treatment based on the positive data from the randomized phase 3 TOGA study that investigated Trastuzumab for HER2 overexpressing or HER2 amplified gastric cancer. One of the reasons why it is so difficult to treat gastric cancer with targeted treatment is probably the co-amplification and co-activation of receptor tyrosine kinases. This is something we see in our lab. You see the plots here. When, for instance, we treat cell lines which are characterized by amplification of MET with a MET kinase inhibitor, or when we transect it with c-MET specific silencing RNA, what we see in quite a short time is that other receptor tyrosine kinases such as HER2 and especially HER3 are upregulated, and this ultimately leads to the resistance against c-MET inhibition. Fortunately, we have at least one targeted treatment that is effective. I am speaking here about the anti-HER2 directed monoclonal antibody Trastuzumab, which prolongs survival in stage 4 gastric cancer with an overexpression for HER2. The overexpression status is determined by using immunohistochemistry. Patients with an IHC score of 0 or 1 receive chemotherapy without targeted treatment. Patients with an IHC score of 3 receive platin and a fluoropyrimidine plus trastuzumab, and patients with an intermediate immunohistochemistry score are usually assessed by in situ hybridization, and if in situ hybridization is positive, shows amplification of the HER2 gene, this group of patients is also treated with trastuzumab. And on the right hand side of this slide you can see the overall survival results of the TOGA study, which show a clear and statistically significant prolongation of the overall survival duration when trastuzumab is added to systemic chemotherapy. Another important question was if, upon progression to a first-line trastuzumab-based systemic treatment, it makes sense to continue with trastuzumab and just change the chemotherapy backbone. This question was taken up by a group of Japanese investigators in the trial which is shown here. Patients with HER2-positive advanced gastric and GE junction adenocarcinoma refractory to first-line chemotherapy with fluoropyrimidine, platinum and trastuzumab were randomized to receive either paclitaxel alone or paclitaxel and trastuzumab. And as the Kaplan-Meier survival curves on the right-hand side show, there was no benefit at all by the addition of trastuzumab. This plot here shows progression-free survival, but also looking at overall survival, no benefit at all was seen in this study. To understand the results of the second-line study better, it is important to talk about heterogeneity of gastric cancer. This slide shows a typical phenomenon, heterogeneous or focal HER2 expression in one single individual tumor. In the center of this slide you see the immunohistochemistry staining of the HER2 receptor, and you clearly can see that in the center of the picture there is a nice membranous overexpression of HER2, while in the left-sided and right-sided peripheral parts of this slide there are parts of the tumor which look quite different. They do not have an intestinal tubular-like growth pattern anymore. They show more a diffuse type growth pattern, and the tumor cells are not or only weakly stained for HER2. It doesn't change when you use in situ hybridization instead of immunohistochemistry. In situ hybridization shows exactly the same picture, with parts of the tumor which show a high amplification of the HER2 gene and other parts of the tumor which show no amplification. This heterogeneity ultimately leads to tumor evolution and development of resistance. This has been shown in several working groups. Here is one example from the working group of Jelena Janjigian at the Memorial Sloan Kettering in New York. She compared pre-trastuzumab gastric cancer biopsies with immunohistochemistry and with in situ hybridization, and she showed that in a majority of patients upon progression to a trastuzumab there was a loss of HER2 overexpression and loss of HER2 amplification. In addition, HER2 resistance defining mutations, like for instance PIK3CA mutations, could occur. We are really dealing here with the phenomenon of tumor heterogeneity and biological evolution during drug treatment in the sense of Darwinian evolution. The result of that is resistance against targeted treatment. Genomic heterogeneity truly is a barrier to precision medicine in gastric cancer. This is work from Daniel Catenacci's group from Chicago. They showed that gene mutations are shared between primary and metastasis in only 58% of their investigated patients, and gene amplifications are shared in only 37%, while in the rest they are restricted to the primary only or to metastasis only. And when they investigated cell-free tumor DNA, this even added to the level of complexity, as they found also alterations that were not prevalent in the primary tumor or in the distant metastasis, and were probably reflecting another level of genomic heterogeneity coming from sites they had not biopsied before. This slide illustrates what we probably have to do in the future when we apply precision medicine to patients with gastric cancer. Relying on the baseline sampling alone will probably not be enough. We need to reassess the tumor upon relapse and even more upon progression to targeted treatment. Probably the analysis of cell-free DNA will enable us to get more information about the genomic evolution of the tumor. The potential value of analysis of cell-free tumor DNA has been nicely demonstrated in a recent Korean publication in the Annals of Oncology. They took samples from patients included in a trial that assessed lapatinib. Lapatinib is a small tyrosine kinase inhibiting molecule directed against HER1 and HER2. And what they saw here is that upon initial response to lapatinib, some of the alterations disappeared, like for instance the yellow one, the HER2 or ERP2 amplification got less. Upon progression, the HER2 amplification in the cell-free DNA came back, but also new amplifications occurred, like for instance FGFR2 amplification and cyclin E1 amplification, which contributed to the ultimate resistance of the disease against lapatinib. This slide illustrates another example from the same publication. Again, upon response to lapatinib, the yellow signal HER2 amplification disappeared, but upon progression, a new signal came up, the blue one, which indicates MET amplification, which is a well-known and typical resistance mechanism against targeted treatment of HER1 and HER2 receptor targeted compounds. One could speculate that precision medicine and targeted treatment against gastric cancer may be more effective in the localized stages when we apply it in the neoadjuvant setting, because in this stage tumor heterogeneity is probably still less pronounced than in the metastatic stages. And this slide here is showing you the example of two studies which investigate trastuzumab or trastuzumab and pertuzumab, another anti-HER2 directed antibody in the neoadjuvant setting of gastric and EG junction tumor. The JCOG, the Japanese investigators, combined trastuzumab with S1 and platin in localized HER2 positive gastric cancer with bulky nodal disease, in their respective studies called the TRIGGER study. And we investigators from EORTC currently perform the innovation study, which randomizes patients with HER2 positive localized gastric cancer or GE junction adenocarcinoma to 3 arms. The one is perioperative chemotherapy alone, the second one is peri-op chemo and trastuzumab, and the second experimental arm is chemotherapy and trastuzumab and pertuzumab. Moving forward to another subtype of gastric cancer, I'm talking now about the genomically stable type of gastric cancer. Interestingly, this molecularly defined type of gastric cancer has a clear association with the diffuse histology according to the old Loring classification, and some of the molecular alterations that are typically found in this type of gastric cancer are mutations at the CDH1 and at the RHOA genes, and fusions between Claudin 18 and RGAP. Interestingly, when we look at molecular differences between early onset diffuse gastric cancer and late onset diffuse gastric cancer, we see that mutations and alterations at the CDH1 level are more common in early onset gastric cancer, while mutations at the level of RHOA are more characteristic for late onset diffuse gastric cancer. Concerning the outcomes of these patients, we see that alterations at the CDH1 level are clearly associated with a worse prognosis, and this is something which all of us who treat patients in the clinics know, that sometimes we have these very young patients with diffuse gastric cancer who have a very problematic outcome, recur early after surgery, and are characterized by very aggressive disease. There may be some hope now, especially for patients with diffuse type gastric cancer, in which an adhesion protein Claudin 18.2 is probably more prevalent than in the intestinal type of gastric cancer. What is Claudin 18.2? It is a member of the Claudin family, as the name suggests. It is a major structural component of the tight junction. It seals the intercellular space in epithelial sheets, and it is typically not expressed in any healthy tissues except the stomach mucosa. In contrast, it is typically expressed in malignancies, especially in gastric malignancies, but can be also found in esophageal, pancreatic, and some other intestinal malignancies. And as mentioned before, a typical overexpression is found in the diffuse type of gastric cancer. A biotech company from Mainz, Germany, developed a monoclonal antibody which is called IMEP362 and meanwhile has the name of Somatic. This antibody is directed against Claudin 18.2. Its major mechanisms of action are believed to be immunogenic by activation of ADCC, antibody-dependent cytotoxicity, and also by activation of CDC, complement-dependent cytotoxicity. In a randomized phase 2 trial with a limited number of patients, it was shown that the addition of IMEP361 to standard chemotherapy, epirubicin, oxaliplatin, and capecitabine, at that time led to a significant prolongation of progression-free survival and overall survival. Interestingly, these good effects seen with Somatic were more pronounced in the diffuse type of gastric cancer, and they were also more pronounced when the level of Claudin 18.2 cellular staining increased. As you can see in the highlighted part of this slide, an overexpression in more than 70% of the tumor cells led to a more impressive hazard ratio than an overexpression in less than 70% of the tumor cells. Based on these findings, 2 globally activated validation phase 3 studies were started. This slide here is showing the design of the so-called SPOTLIGHT study. that aims to include 550 participants with first-line advanced gastric cancer and a claudin 18.2 positivity in more than 75% of tumor cells. Patients will receive either modified FOLFOX as a control regimen or modified FOLFOX and Somatic as the investigational regimen. The primary endpoint will be PFS, secondary endpoints will be overall survival, overall response rate, duration of response, health-related quality of life, and others. Coming back to the different biologies of gastric cancer, we are moving now to the interesting subtype of microsatellite instability, MSI gastric cancer. This molecular characteristic can be assessed on the genetic level, for instance by performing polymerase chain reaction, but it can also be assessed on the immunohistochemistry level by staining for one of the DNA mismatch repair enzymes such as MLH1. The loss of MLH1 in this example here characterizes the status of deficient DNA mismatch repair or microsatellite instability. Microsatellite instability gastric cancer is characterized by a high mutational burden. It occurs more frequently in the intestinal compared to the diffuse subtype of gastric cancer. Its location is more often in the distal than in the proximal parts of the stomach. You can see that at the level of the esophagogastric junction, microsatellite instability is an extremely rare finding. Interestingly, this subtype of gastric cancer is more prevalent in females than in males, different from all the other subtypes, and the median age of these patients is the highest compared to the other subtypes of gastric cancer. There is a very interesting observation from a large cohort of the Yonsei University gastric cancer patients, which tells us that after resection alone, patients with microsatellite instability gastric cancer have a better prognosis than patients with microsatellite stable gastric cancer if they receive no chemotherapy. Looking at patients who received adjuvant therapy, you do not see a difference between the prognosis of MSI high and MSI low or MSS patients anymore, which suggested that there is minor or even no benefit associated with adjuvant chemotherapy for MSI high gastric cancer patients. A confirmatory observation was made by the MAGIC investigators. They investigated the prognosis of patients who received surgery alone or perioperative chemotherapy, and as you can see on the left-hand side of this slide, the gray group were the patients who received surgery alone and had microsatellite instability gastric cancer. They really had the best prognosis of all patients, while the orange group had the worst prognosis, and these are patients who were diagnosed with microsatellite instability gastric cancer and received perioperative chemotherapy. A word of caution, this is a retrospective analysis on a limited cohort of patients, but again, it tells us that potentially perioperative chemotherapy is of no benefit for patients with MSI high gastric cancer. It could even be detrimental. And when we move to the right-hand side of this slide, we see survival curves from a Korean study, the CLASSIC study, which investigated post-operative chemotherapy versus surgery alone. And again, you see an interesting phenomenon. The MSI high gastric cancer patients, these are the red curves, have the better prognosis, but with no benefit coming from adjuvant chemotherapy, while the blue curves, which stand for MSS, microsatellite stable gastric cancer, had a worse prognosis in general, but still a positive benefit from adjuvant chemotherapy with oxaliplatin and capecitabine. When we go back to the biology of microsatellite instability gastric cancers, we know that in these patients, parts of the DNA mismatch repair complex are deficient, leading to deficient nucleotide repair and to an increase of mutations. More mutations means more production of antigen precursors, which after intracellular processing can be presented as neoantigens via HLA molecules at the cell surface of the tumor cells. All this can render the tumor cell more recognizable for the immune system, for instance, for killer T cells. Making tumor cells more recognizable by the immune system is a prerequisite for the efficacy of immune checkpoint therapy. As you know from the famous immune checkpoint cycle published by Janet Nalman in 2013, we have several levels at which we can enhance the immunity against tumor. Two typically applied mechanisms today are the immune checkpoint inhibitors directed either against CTLA4, which enhances the training of the T cells at the level of the lymph nodes, or the application of immune checkpoint inhibitors directed against the PD-1, PD-L1 axis, which activates CD8 positive T cells at the level of the tumor. What do we know about the efficacy of immune checkpoint inhibitors in the clinic? One of the most important studies has been published recently in Lancet. This study compared pembrolizumab, a checkpoint inhibitor directed against PD-1, with weekly paclitaxel as a second-line treatment for patients with advanced gastric cancer. One can see that the overall efficacy of pembrolizumab as compared to paclitaxel was not as good as expected. The overall hazard ratio, which means the reduction of the risk to die, was only 0.82, which corresponds to an only 80% of risk of reduction with the application of pembrolizumab as compared to paclitaxel. In the early phase of treatment, even detrimental effects were seen if pembrolizumab was given instead of paclitaxel. But when you go to the right-hand side of this slide, you can see that a subgroup that clearly benefited from receiving pembrolizumab instead of paclitaxel was the group of patients who presented with MSI-high tumors. This has also been demonstrated by a Korean group of investigators. They recently published in Nature Medicine that patients with MSI-high tumors, but also patients with Epstein-Barr virus-associated tumors and patients who present with gastric cancers with a high expression level of PD-L1 are those who have the best chances to benefit from a PD-1 immune checkpoint inhibitor like pembrolizumab. PD-L1, the programmed death ligand 1, which is part of the PD-1, PD-L1 axis, can be expressed either on tumor cells or on immune cells of the tumor stroma. Currently, different groups try to define which form of overexpression is most predictive for the effectiveness of immune checkpoint inhibitors. PD-L1 overexpression at a smaller level does not seem to be an optimal biomarker for predicting the efficacy of pembrolizumab. But when the level of overexpression is augmented from a combined positive score of 1 to a combined positive score of 10, the enrichment of patients who benefit from pembrolizumab blue curve as compared to paclitaxel red curve is already better. This is a figure from the Lancet 2018 publication, and it represents an exploratory post hoc analysis. So this is not definitive knowledge, but this plot suggests that a combined positive score for L1, which represents the combination of tumor cell staining and stromal immune cell staining with a threshold of 10, might be an interesting biomarker for defining patients who benefit from PD-1, PD-L1 blockade in the future. Taking a short look at the last of the 4 subtypes of gastric cancer, the EBV positive, Epstein-Barr virus positive type of gastric cancer. I have mentioned it already before when we saw results from Korean investigators who reported that patients with EBV positive gastric cancer are more probable to benefit from treatment with the PD-1 inhibitor pembrolizumab. Indeed, the EBV positive type of gastric cancer is often exhibiting a typical lymphoepithelioma-like carcinoma morphology with infiltration of many immune cells. Also, in this subtype of gastric cancer, PD-L1 and PD-L2 overexpression is frequently found, and immune cell signaling is activated together with some other molecular alterations which are typical for this subtype of a very immunogenic gastric cancer. Let me summarize. The personalized treatment of gastric cancer is not yet here, but it is hopefully emerging. We already have HER2-targeted treatment in HER2-positive gastric cancer available with the first-line approval of Trastuzumab. Unfortunately, other receptor tyrosine kinase-directed drugs failed thus far in randomized controlled trials. The excessive gastric cancer tumor heterogeneity and evolution complicates targeted treatment in gastric cancer. And I would speculate that early stages applying precision medicine to neoadjuvant treatment may be the better scenario for a precision medicine approach, especially in diffuse type gastric cancer, but not limited to that. Claudin 18.2 is an emerging target, and randomized controlled phase 3 studies are ongoing. Results are awaited within the next years. Finally, and last but not least, the MSI high, the PD-L1 positive and the Epstein-Barr positive gastric cancer subtypes are probably good candidates for immune checkpoint inhibitors. I thank you very much for your kind attention.